let's say there are 20 fields of science, right? I don't know how many departments there are at MIT or whatever, but let's say there's sort of 20 buckets, neuroscience, condensed matter physics, astronomy. And let's say within one of those fields, there's maybe 5 or 10 of these kind of like Hubble Space Telescope level, like kind of key observational capabilities or perturbational capabilities. Well, so 20 times 5 is 100. There's like a few hundred things that sort of should be these things in the gap map that could be addressed by this kind of FRO-style approach. And let's say each one of those costs tens of millions of dollars, okay? It's a lot of money. It's like on the order of like 10 billion, 15 billion to like take out the gap map as like a set of gaps in science. That's like large, but like it's less than the National Science Foundation spends in a year, way less than the NIH spends in a year. It's way less than industry R&D budgets. It's way less than individual programs.
It's like a rounding error in the Excel sheet of our economy.
Today we're joined by Adam Marblestone, a scientist and strategist whose career spans neuroscience, nanotech, and synthetic biology. Adam is the CEO of Convergent Research, where he creates focused research organizations, or FROs, startup-style teams built to tackle scientific bottlenecks that universities and companies can't solve alone. In this conversation, we discuss past breakthroughs in neuroscience and how FROs can reshape the future of how science gets done. Please welcome Adam Marblestone.
I was already pretty obsessed with this notion of like broadly applicable technology platforms, so I was very interested in biomolecular nanotechnology and DNA origami, and could you use DNA and other biomolecules to sort of self-assemble much more complicated multi-material kind of microchips. So I started out grad school working on that, and I guess it's all a little bit related. I eventually concluded kind of midway through grad school that basically we were never going to compete with Intel, right? It was like I didn't see a way to get that 20 or 30 person team or even larger team really working together on building what George Church was my PhD advisor. I was also working with William Shi and Peng Yin and other people in the nanotechnology space, but we had this concept called nanometer to centimeter, NM to CM, where we were interested in sort of fully addressable microchips. Could I have a unique DNA strand at each location on a microchip that would allow you to uniquely, deliver a certain whether it be nanoparticle or DNA origami or some other component to that location on the chip. But we kind of concluded there isn't like this economic driver for it. And so then I had the kind of hard pivot into neuroscience.
What was it like? I think, we've had George Church actually on this podcast.
Yeah,
and
nice.
I think he's kind of like a quintessential scientist. He looks like a scientist, you know? Like I think, and he talks like it's like this, he's almost like a mascot for science in a way.
Yes.
What was it like working in his lab?
It was fantastic. I mean, it really was. Every time I would come to him and I would say, I'm thinking about doing X experiment, he would say, okay, great, but here's some ideas for how you might make that 1 million times more scalable or more ambitious. What if you multiplex this by a factor of 1,000? What if we work with this company? What if we did this big collaboration? And so that was very much encouraging of my like trying to think on a scale that wasn't really something that I wasn't that fantastic an experimentalist. Maybe with different training or something, I could have gotten better at it, but I very much felt limited by what I could do individually. George was like very encouraging of big visions, bigger projects, bigger collaborations, and he had a ton of insight into the experiments. He was proposing lots of specific stuff. He's very, very in the weeds and really prioritized working with the students, even though he had like 100 people affiliated with the lab. Like the thing he would prioritize rather than meeting with like
he really scaled his lab.
Yeah. So I mean, and those were not all like directly under his supervision. Some of those were affiliates of different kinds, but he really would prioritize like meeting with that new grad student like over some important business meeting or something like that. So it was really fantastic in those ways. It was also so cross-disciplinary that we sort of had to invent a lot of stuff for ourselves, right? Basically, depending on which postdocs were floating around and what people were interested in the lab, it was just a completely different lab, even from like some number of months to some other number of months. It wasn't even really a lab that was doing that much in neuroscience. And so the angle that we started out with in neuroscience was, okay, we were interested in these transformative tools, platforms. How do you make neural recording 100 times more, 1,000 times more cheaper, scalable?
And when you say neural recording, do you mean like brain computer interface, or like taking a dead brain and figuring out what the connections are, or like what
it ended up being both, and I've ended up over the longer term having even more interest in the dead brain circuit mapping, which maybe we can come back to because one of the first FROs is working on that. But at the time, this was like around 2012, 2013 era. And this was the beginning of what became the Obama administration Brain Initiative. And George and a number of other people had been involved in sort of the precursor of what became the Obama administration Brain Initiative was actually a white paper about something that was called the Brain Activity Map Project, or BAM, which actually came about from meetings that the Kavli Foundation had organized, bringing together, they had funding in nanoscience and they had funding in neuroscience, and they brought together nanoscientists and neuroscientists and said, okay, what are the big kind of grand challenge problems? And some of the neuroscientists came up and said, well, right now we can record the electrical signals, the kind of real-time activity traces, from the order of sort of hundreds to thousands of neurons. Even a mouse brain has 100 million neurons. And so there's a big architectural question of how would you sort of scale it up. And so the nanoscientists were saying, oh, well, maybe we could contribute to this. Ultimately, those discussions actually in a really meaningful way led to what became the Obama administration Brain Initiative, billions of dollars in funding for sort of neurotechnology tools, techniques, technologies for neuroscience. From the perspective of where I was sitting in the Church Lab with the sort of biomolecular nanotechnology and physics background, not really as an actual practicing neuroscientist. I had never actually stuck electrodes into a rat brain or something like that. We were thinking about it in terms of whether you could use DNA sequencing initially. This was the initial question of whether you could use DNA sequencing as a way to read out information from the brain in a much more kind of scalable way. And this was based on a kind of thought process or analogy that had happened in many other fields, where first there was the Human Genome Project, and then George and a bunch of other companies and technologists reduced the cost of sequencing the human genome from, on the order of like $1 per base pair, so billions of dollars per genome, down to, by a factor of hundreds of thousands and then millions, down to sort of sub-$1,000 genome. But when they had that technology of DNA sequencing, people then started applying that to sequencing other molecules. So people were looking at RNA sequencing. Single-cell RNA sequencing was sort of getting started at that time, which has been a huge revolution in bio. Looking at the 3D structure of the genome, looking at all sorts of other things you could read out with DNA sequencing. So we were interested in, could you read out neural activity with DNA sequencing? And that would be a pretty unusual concept for how you record information from the brain, because normally, in order to get sort of real-time, high-speed information out, you basically need the information to be transmitted in real time out of the brain. There isn't any kind of internal to the brain, like memory storage for the recorded signals. It's like you have an electrode, and then it basically gets like kind of digitized kind of after it's come out or on the way out. Or you have like light-based optical fluorescent methods of recording, monitoring the neurons. The light basically goes out and hits a camera. And so here the question was, could the individual neuron cell like record some activity trace or ticker tape of its neural activity into DNA, which then at the end, you would sort of fix or freeze the brain, and then you could record. Now you would have decoupled, right? You could read out the information that might have been recorded initially into the DNA substrate very quickly, but then you could take as much time as you wanted to read it out from the different regions and sort of slice it up and read it out slowly. So having like an internal memory store in the form of the DNA would have totally changed the kind of bandwidth question of how to get the information out and digitized. So we did some very early and kind of almost embarrassingly preliminary experimental work of like, can you record any kind of like time varying signal into DNA, and any kind of signal that isn't really itself DNA, can you record that into DNA? So we had these DNA polymerase, DNA coupling enzymes just in test tubes, and depending on the concentration of different ions in the test tube, we were working on calcium, and we got sort of weak results and initially kicked off some collaborations to do recording of the calcium ion concentration, because that's something that's relevant for neurons, that their calcium concentration changes when they fire. But there were other ions like manganese that could much more easily affect the DNA polymerase. So we would make these kind of time traces of high manganese, low manganese, high manganese, and then the DNA polymerase would be copying as this ion concentration was changing. And could we then read out from the sequence that it went high, low, high, and so on. So it's sort of very preliminary, just few bits of information of kind of recording ion
So
concentrations.
what happens to DNA in the calcium-enriched environment?
Yeah, well, what was happening is that the DNA polymerase, the copying enzyme, has a certain error probability. So normally, and when it's talking about copying the DNA of a human cell or of bacterium, the error rate is incredibly low, and it has lots of proofreading that's involved in that. But there are actually some DNA polymerases that are involved in other biological processes, like if there's some radiation that actually mucks up a piece of DNA, there's a polymerase that needs to be able to copy past the kind of chemical lesion that gets created, so these lesion bypass polymerases, and those ones can have much higher error rates. They just want to put any base of DNA in so that they can get past that lesion and that the process can keep going. And it turned out that some of those lesion bypass polymerases were naturally very ion sensitive in terms of what is the probability of them incorporating different bases.
A particular base. Yeah,
because basically the ion sort of fits in this polymerase active site, and depending on the shape of an A versus a T, for example, it might miscopy. It's supposed to put in an A and instead puts in some other base. And so the ion can affect the sort of probabilities of each base being incorporated. And so we could have these ion-dependent error rate DNA copying reactions. So that was pretty cool. And then later, other people really have figured out, I think, better, more interesting ways of recording some sort of time-varying cellular signal into a molecule that you can read out later. There's been recent interesting work from Adam Cohen's lab and Ed Boyden's lab and Kirill Plakayevich's lab on recording into these protein filaments of self-assembling proteins that go end to end. And they can actually have sort of patterns along the length of these protein filaments that you could read out by microscopy or by something else that depend on sort of the history of events in that cell. So anyway, we were thinking a lot about that. But what that really was actually just for me, it was a kind of a gateway drug, I guess, to thinking about neuroscience. And so what I ended up doing is kind of thinking, okay, well, this ticker tape line of experimentation, what it tells me is that the field of neuroscience is not necessarily in a global maximum, basically, of the technologies it's considering for doing, whether it be neural recording or other types of neuroscience kind of observation. So we ended up doing a lot of kind of road mapping, getting together different physicists, chemists, other people from different fields, and sort of road mapping what are all the different ways that you could do neural recording, combination of ultrasound plus optics, optics plus different biology, DNA plus other things, ultrasound plus electronics.
What was like back then the default, like the best idea back then versus what turned out today to be like what people are doing today?
I mean, it's very much still evolving. If you look at like Neuralink or major companies, I mean, that's still an electrode-based approach, and it's really technical improvements in the technology of just sticking a wire.
That was like
right
the
near
default kind of like old school approach that everybody's trying to come up with something else.
Electrophysiology, and there's always the question of how much can electrophysiology scale, but in the past like 10 years, actually, I mean, so in terms of the human brain-computer interface and sort of maybe what are the most advanced things, it's still mostly electrophysiology. They're using tiny kind of wires where they can fabricate very, very little bits of metal on very flexible polymers and kind of insert those electrodes with a little sewing machine, they call it, allow that very little physical volume and very flexible wires, but it's basically just wires going next to the neurons. Even in like basic neuroscience, academic neuroscience.
I've never understood exactly what that
arrays.
means. Like if you have neurons and they're like 10 microns in size, how many neurons are actually talking to one wire? Like if you're sticking the wire in, what's actually happening? The wire must be like, 100 microns.
Yeah, so there's a thing called spike sorting where if you have this sort of extracellular electrophysiology, there may be anywhere from a few to maybe like potentially dozens of different neurons near that electrode that it in principle can see, and then they will have different kind of extracellular voltage patterns that you may be able to see depending on depending on basically how fine of sort of analog resolution and how you're actually recording faster than just was there a spike or not. You're actually seeing something about the spike shape. And so the neurons will have a different spike shape and spike amplitude. And you can kind of sort from a given electrode.
So that's actually a good thing, because then you get many neurons to a single wire.
And then it's even more so also if you can oversample, if you can have even more. There's also places with something like neuropixels is this thing that's been created since then, which is a sort of tightly packed array of electrode pads on a single like silicon shank. And so that thing can, in some cases, can kind of almost like oversample the number of neurons. And so you're actually seeing like a spatial pattern of a given neuron is going to show up on multiple different pads to different degrees and sort of spatially localize a little bit where that neuron is coming from. But so a lot of the advances in what's been practically used is still, I would say, extremely good technical work scaling up electrode-based recording. There's also been a lot of progress.
How did that
optical.
start? It's literally people putting a wire in a mouse brain, like just sticking it.
Yeah. I think it goes back, maybe back to
Stabbing
Helmholtz.
it.
And
Okay.
I think the downside is that it's mostly damaging, and I guess like the top neurons where
it makes
the
it
needles
really
would go through, they kind of damage most of it, and it's like not that specific from area where you can.
And so like with the neural sewing machine with Neuralink, you have to be extremely careful not to hit a blood vessel with that sewing machine.
I wonder if the
inserter.
real solution is like to implant a baby brain with all the electrodes and then let the brain grow around them or something.
Yeah, there's some thought to that, and there's another company called Science that Max Hodak is running. He was one of the Neuralink founding team members, and which is doing some experiments basically on cellular electrodes. So can you basically have a neuron send its axon in, make synapses?
Right, so it's like a cell-to-cell.
It's a cell-to-cell contact,
and then it's like optogenetics to get the like fluorescent neurons.
Right,
to get
you could stimulate it, or you could basically have neurons where you don't know exactly the kind of matrix of transformation that these intermediate neurons are telling you, but you're still getting a lot of information in like a less invasive way. So a lot of there's a huge momentum in the field. There's also optical methods, which we haven't really gotten into that much, but they require basically a camera to look at each individual neuron firing with changes in fluorescence. Due to the calcium changes or things like that, voltage changes. And so
I've seen one recently from Thor Labs that was quite amazing. They would mount this micro camera on top of the mice, like with the.
But that's when you put a window
surgery.
like a plastic window.
Cranial surgery.
You can put a little window. There's something called the mini scope. So there's a tremendous amount, and I think it's all great stuff. But there's a tremendous amount of kind of like momentum of techniques that are actually workable and give actual good scientific results. And of course, the molecular ticker tape idea with this DNA recording, like it's still to this day not really given any neuroscience results, right? So it's not something that's that usable, but it's like this huge technology push. Maybe it's coming in that direction. Other things that we were thinking about when we were road mapping this, we and a couple other groups, right around that same time, there was a paper on something called neural dust, which was about little piezoelectric chips that would respond to ultrasound. That basically ultrasound would come into the brain and reflect off this little piece of piezoelectric material, and that piezoelectric material, its kind of like stiffness would be modulated by the local electrical signals that it was picking up from the neurons. And that would change how much the ultrasound could sort of backscatter. And so you can use ultrasound, and ultrasound has a lot of advantages in terms of the trade-off of sort of spatial temporal properties that it has. So like with light, most wavelengths of light that are interesting for looking at changes in neural activity.
They're not going through the skull.
They don't. Yeah, they kind of they scatter a huge amount in the opacity of the tissue, and or they get absorbed a lot, and so it's hard for the light to really fully penetrate. It's certainly for it to penetrate on like a linear path. Ultrasound, it has trouble going through the skull, but if you can get on the other side of the skull, emit and detect the ultrasound from inside the skull, not necessarily inside the brain, but from inside the skull, the ultrasound at wavelengths that are sort of roughly the size of a cell can penetrate relatively undisturbed through centimeters of brain tissue. And so actually the trade-off of being able to go centimeters deep while having sort of roughly cell-sized spatial resolution is really powerful, but then you have to somehow transduce that ultrasound signal. So we were also thinking a lot about, should you use neural dust? Should you use a combination of optical transducers that would then couple into ultrasound in different ways? Ultrasound can affect the phase of the light, or you can have photoacoustic effects that will generate ultrasound with light. And there's been a lot of great work since then. Anyway, so one of our focus research organizations,
just recording like a neuron firing with ultrasound, like, is that actually happen?
there is a field that's also, I think, kind of mostly emerged over the last 10 years called functional ultrasound recording. And that's actually, it's mostly not looking directly at neurons, it's looking at microvascular changes in the blood vessels as the neurons ask for more oxygen. This red blood cells will flow faster or slower, or you'll have changes in oxygenation and so on. And so functional ultrasound can see some of this.
So that's like fMRI, but ultrasound.
It's more like a micro fMRI, but that can be done with ultrasound already. So anyway, so one of our focused research organizations now, sometime later, is using this ultrasound-on-a-chip technology. So it's a chip with a MEMS chip with lots of little cantilevers, basically, that can emit and detect ultrasound as a phased array. So they're using that actually in humans to do functional ultrasound imaging. But so anyway, we were thinking a lot at that time about what's the kind of global maximum of the field would be, maybe molecular ticker tapes, combinations of optics and ultrasound and biology and other things, devices. Most of the momentum in the field has still been push on these electrodes, push on this calcium imaging. But this comes back a little bit to the thing about the focused research organizations, is sometimes you need like a different organizational structure and a kind of concertedness of resources to actually shift. Like, we need to do a big technology project. It's going to be a while before that big technology project incrementally advances an individual neuroscientist's understanding of the brain and ability to publish papers about the brain. Right. One way we sometimes think about it is neuroscientists are not chip designers, right? If you're a neuroscientist, you need to get neuroscience results, not chip design results, fundamentally. And so how do you then develop the technology that neuroscientists need? You somehow need to get a team of chip designers working in service of neuroscience.
It's very much the type of thing happening at Neuralink, I guess.
That's right. Yes, bringing a larger engineering team. This comes back to all this stuff, Convergent and all this stuff, is, how do you have these cross-disciplinary engineering team in service of a scientific field when doing that work isn't necessarily incentivized within the field itself to be developed?
You have this tool on your website called Gap Maps, which reminds me of like a supercharged version of something I used to do, just like write notes about what are the problems.
Yeah.
I used to think about this a lot, like I had more free time, so I was like, what are all the science problems,
Right.
And I would love to go to a Gap Maps conference with somebody giving a talk on each one of there's like probably 100 items on that.
Yeah, there's I think on the order of 100 of what we're calling the gaps, and there's more than 100 of what we're calling the sort of fundamental capabilities that are like potential partial or complete like solutions to the gaps that may be developed to different degrees. I mean, this one is also not all problems in science. Like, we don't have on there, just understanding, the learning algorithm of the cortex or something. That's like a science problem that has a theoretical component, an experimental component, and kind of iterative discovery needed to get to it. And there are, of course, many scientific questions that haven't even been formulated yet, right? And a lot of what people are doing when they're scientists is formulating a new question. So we're not at all trying to say with the gap map that this is all the questions, but we're trying to make a stab at saying this is kind of like captures some of the variance, meaningful part of the variance of these sort of roughly FRO-shaped projects where there's like a well-defined, legible tool system, dataset platform, systematic project or program that's just like, if it had resources and leadership and execution, that could be really accelerating of something that is otherwise like not very well served. And so if you think about the fundamental capabilities in that way, FROs or like ARPA, DARPA type programs, these relatively legible ones, and relatively kind of medium-sized chunks of ones. So we call it the fundamental development gap map as opposed to just like the science gap map.
Yep.
So it's not all science, but I think it shows you, right? Sometimes I like to think about it as it's a very large but not insurmountably large like set of problems. So if you imagine that each one of these things, let's say there are 20 fields of science, right? I don't know how many departments there are at MIT or whatever, but let's say there's sort of 20 buckets of like of science at large. Neuroscience, condensed matter physics, astronomy, and let's say that there are, within one of those fields, there's maybe on the order of 5 or 10 of these kind of like Hubble Space Telescope level, like we have to be able to record the neural activity, and we have to be able to map the wiring, and we have to be able to see the molecules, and we want to know how the system develops, and there's some kind of finite list of like kind of key observational like capabilities or perturbational capabilities. So 20 times 5 is 100, right? So order of magnitude, there's like a few hundred things that sort of should be these things in the gap map that could be addressed by this kind of FRO style approach. And let's say each one of those costs tens of millions of dollars. Okay. It's a lot of money.
Over what period of time?
That's tens of billions.
Half decade.
Kind of
it's tens of billions. Yeah.
Yeah.
It's like on the order of like 10 billion, 10, 15 billion to like take out the gap map as like a set of gaps in science. That's like large, but like there are individual people who have that.
It's like a rounding error in the Excel sheet of our economy.
It's less than the National Science Foundation spends in a year, way less than the NIH spends in a year. It's way less than industry R&D budgets. It's way less than individual programs.
What's the top of all the gaps? Which one do you think is the most likely to be kind of like an OpenAI, where if they actually come up with something like a ChatGPT moment comes out of one of these things, it's going to have this kind of like insane, venture capital boom and like people on TV losing it about whatever. Oh no, we've mapped the brain. Now everyone's going to lose their job. Kind of like a response.
I mean, it's hard. It's kind of hard to beat AGI. I mean, some of the things that we are. Broadly, I still think that neuroscience is one very big one. If we could really like program developmental biology, right? If I could instead of having this choice of like my plant-based meat or my like animal-based meat or my like cultured cell-based meat, which costs $10,000, I could have like just grow a steak or something like that. I mean, that would be pretty great. But actually, one thing that we're doing that I'm particularly excited about. I would say it had more of a catalytic role because it was an existing open source project, but we've converted it into a FRO and gotten it a lot of resources, is this project called Lean, which is a theorem-proving language for math. So if instead of writing your math on pencil and paper or in LaTeX or some sort of informal language, you write it in this computer language called Lean, and you can click verify, and if your conclusions follow from your assumptions, and the proof that you've written is correct, it will say, yes, it's correct. And if not, it will say, well, there's something messed up at step 5 here. This doesn't follow.
It's like what, math like T9, and what is it, autocorrection? So how in the messages here will be, is your math correct or not?
It's a verifier. It's an automatic verifier.
Like a math compiler.
Yeah, it's like a compiler and can tell you if your code doesn't compile.
Yeah.
And so I think this one is poised to be super important. So for one thing, a bunch of companies have already started up saying we're going to make mathematical superintelligence. Basically, we're going to use this as a reinforcement learning signal, right? So I'm going to try to generate some math, and if it's true, then I get a reward, and if it's not true, I don't. And of course, there's the question of, can it formulate interesting conjectures and so on, or will it be able to learn that on the way to proving some well-defined result that you've already formulated? But sort of, can you automate math? And if you can automate math, I think you can also make software in a very different way. So instead of saying just vibe coding, right, what people are doing now, you actually say, here are the provable properties I want the software to have. I want it to never have this vulnerability where someone can hack into the memory in this way. I want it to be provably secure in these ways. I want it to be provably optimal in these ways. And then you can synthesize code, and not only synthesize code and does it compile, but you actually have a mathematical proof that code satisfies that spec, which could include a security spec. Can you make it impossible basically to have a cybersecurity vulnerability because it's actually mathematically provable that it's impossible to hack into this system, right? So I think that this formal methods and formal math is one of the areas where it could be very big. That's not totally distinct from the AI thing, but I think that could be a pretty big deal. Yeah, for a relatively short time scale.
One of the ones I saw on there was you have a couple of kind of like greatest hits ones on there. So we like, joked about like, okay, if you make a list of all the discoveries that would win the Nobel Prize for sure,
superconductivity,
it's like room temperature, superconductivity, low, cold fusion, low energy nuclear reactions. I think one that's on there that maybe it's debatable if it would win the Nobel Prize, but like the ability to make non-sequential polymers, so basically like an artificial ribosome.
Yes, love that one.
It's what do you think? Like, have you seen anything interesting in those areas?
We would like to do projects in all those areas. So we have proposals for roughly FRO-shaped projects in, let's say, new fabrication methods that would allow you to screen through a larger number of candidate superconductor materials or other quantum materials and assay them in a more high throughput way. So sort of like a self-driving lab for discovering superconductors. But that could be based on things that are not just setting up robots, but like lithography systems that would let you multiplex your experiment on superconductors more, and also you
have like pixels with different materials, and then you can test them at high
So
scale.
there's some work sort of towards that direction with the Acceleration Consortium, and U Toronto got like $200 million or something from the Canadian government to sort of do self-driving labs tied to materials. But I think that even there's some FRO-shaped projects, kind of discrete projects, including one that's been proposed on AI that gives you not only what the predicted properties of a new material are, but the synthesis instructions.
Right, that's critical.
So retrosynthesis for materials, complex materials. I would love to do something on artificial ribosomes, not necessarily cold fusion per se, but there's other people that have reached out to us on many body quantum phenomena and things that quantum nucleonics is one thing that people are talking about. Of sort of, is there some scientific basis of sort of studying the things in and around nuclear interactions that might have anything to do with cold fusion? So I think there's stuff in material science. I think there's stuff in superconductors. Artificial ribosome definitely.
You've thought a fair amount about artificial ribosome because it overlaps a little bit with your past work.
It comes from the biomolecular nanotechnology stuff. And can you one question we thought about is there's sort of different versions of the artificial ribosome, right? Actually, DARPA just put out a request for information on something that sounds a lot like a one-dimensional artificial ribosome of sort of sequence programmable multi-material like polymers. It would be amazing if DARPA could fund something in this area, and then maybe we could spin up a FRO. But
so maybe it's good to define the ribosome is the machine in the cell that reads DNA and generates proteins, which are non-sequential polymers, meaning you can decide at each position what's in the polymer, versus like a plastic would just has the same thing over and over, or maybe like ABA or ABBA or whatever. You have this completely programmable thing, and then it can take all kinds of shapes and functions.
This thing basically implements the genetic code. So these triplets of DNA nucleotides, there are 4 at each possible nucleotide position for DNA, which then gets made into RNA. And so you have 64 different sort of possible codons of these triplets, and then each one of those codons goes into one of 21 amino acids, depending on some non-standard amino acids and other things. But basically, it converts these triplets of DNA of RNA code into a equivalent chain of 20 or so building blocks that are not nucleic acids. They're these amino acid building blocks, and the amino acids can also be modified to be non-standard. They have these groups sticking off that is the sort of variable group, where you could make unnatural amino acids. Anyway, so that's what, like, in every one of your cells you have this like polymer synthesis machine that is reading the RNA tape and synthesizing the corresponding protein tape. So one question is, how general of a machine can you make a ribosome-like machine that reads something other than RNA and translates into something other than protein? But the other generalization of it that I think is super interesting is 2-dimensional and 3-dimensional versions. So that actually then becomes like a 3D printer, basically.
Right,
so the ribosome is like a 1D printer controlled by a tape. Can you make a 3D printer that has these covalent bonds? So it's not necessarily atom by atom construction, as people sometimes associate with sort of Drexlerian nanotech, right? It's sort of think about moving one atom at a time.
It's like molecule by molecule.
Yeah, it's like one amino acid size building block roughly at a time. But can you do that with covalent linkages along a kind of 2D lattice versus a 1D chain? Could you do that in a 3D lattice? And then would that be controlled by a tape, or would that be controlled by light?
Is your
or,
intuition that this kind of thing would
heat
be bottoms up or top? Like, is it like a chip that does it? Is it a top down like 3D printer type machine, or is it more like nanotechnology, like what we see with the ribosome is kind of at the scale of the thing it's building?
I think we just need research on all of these things. So sometimes the thing that's tricky about this field is that it tends to be like all or nothing. People are like, oh, this is like totally impossible or bad or just never going to go anywhere, against the laws of chemistry or something to make a 3D ribosome style printer. Or they say, well, this is going to be the future of manufacturing and everything is going to be made on your desktop with this. And I just think it needs to have a research field for each of these things. So I think there should be a research field of sort of molecular chips, which is kind of going, well beyond patterning, silicon, germanium, et cetera, with dopants, but has, which protein do you put at each location, or which DNA origami you put at each location. That's kind of more macro. There's actually some stuff that has spun out of some of the expansion microscopy work where it's an MIT spin-out called Irradiant. Dan Oran has run expansion microscopy in reverse, essentially, to sort of use light to pattern in a hydrogel and then shrink the hydrogel. And now all the things you patterned in are closer to each other. So there's kind of like a chip or 3D chip question. There's like a 1D ribosome, 2D artificial ribosome. There's like a self-assembly question, kind of building on DNA origami. I just think that these things are all like way under-invested in. It's still kind of like this niche field. It's still a little bit far from applications. But I would like to see like really heavy investment in that, kind of in the same way that a lot of fundamental physics and fabrication stuff came out with the motivation of making quantum computers. Here we have a less concrete motivation of exactly what it is that you make with nanotechnology, but it would be great to have DARPA level programs, in molecular 3D printing. Yeah.
So why is it convergent?
Okay, so convergence, this idea of convergence, I think of it in a few different ways. First of all, I guess in like the early 2000s, the National Science Foundation actually, and it still kind of exists today, has this notion of like cross-disciplinary or technological convergence. There's something I read like a long time ago. I was a weird high school student, but I read something called Nano Bio Info Cogno Convergence, was like a white paper.
All the fancy words.
That was put out. And so there's kind of a technological convergence. But I also think what we're doing at Convergent Research, which we can get into with these focused research organizations and sort of our model of organizing projects, it's very much inspired by deep tech startups. So let's say 20 or 30 people all converging together, working together on a single tool, system, data set, product. That might not be a traditional like venture backable, consumer type product, but it's all working in an engineering sense on a single thing. Whereas actually most research I think of is really all about optimizing for divergence. Like your PhD thesis has to be different than my PhD thesis, and that's a good thing because we invented, discovered different stuff. But convergent research is like how you get a team to converge on building something. Maybe there's even a third notion of it, which is like how do we converge on important problems, bottlenecks, pieces of infrastructure that sort of go beyond a single person or effective fields. How do a field kind of converge on the roadmap for what it needs to build infrastructure-wise? Did
this come out of a frustration that you had in your own research? Or is this something you were thinking about like even before you started doing research yourself?
It did. And I can come back to it, but in a lot of different research I've done, but it was particularly true seeing different parts of neuroscience. I felt a need for sort of system building or infrastructure building projects where it wasn't really about the idea or about the novelty of it. It was about building some functioning, I think of maybe the canonical example leading to the focused research organizations is faster, cheaper, better brain circuit mapping, so connectomics. But I think it's also true in other areas, nanotechnology, other areas where you need to build this technology platform, but that technology platform is not really a commercial product. It's really something that folds back into research itself. You could think about the Hubble Space Telescope or many other types of big science infrastructure projects. Those are not really a feat of discovery. It's a feat of coordinating all the different people and disciplines and expertise and incentives to actually get the Hubble Space Telescope built and put in space. And I was just very interested in that as applied to neuroscience, and often feeling like as an individual researcher, there was only so far I could go. But if there was like a 20-person team or something that was well-aligned incentives, we could get much farther. And that frustration, seeing that actually on both sort of the academic side and on the, let's say, long-range startup side, that frustration ultimately has led to my recent obsession with focused research organizations. Yeah.
Is that the term you came up with?
Yeah, so my colleague Sam Rodriguez and I, if I now sort of flash forward a little bit, having thought a lot about neural recording and kind of interacted with an interesting group of colleagues, so Ed Boyden here at MIT and my friend Sam Rodriguez, other people, kind of trying to think about this neurotechnology development in this sort of, Ed would call it working backwards from the problem, right? What are the different modalities, physics, chemistry, et cetera, engineering, that you need to bring to the neuroscience, technology problems? So we sort of had a community thinking about that. And there was a point where I felt like there were some of the big conceptual breakthroughs had occurred. And actually, so we're in this like Tanaka conference room right now. I think Tanaka was one of the people that was involved in this idea of expanding and contracting hydrogels. But so Boyden's lab had this breakthrough on what's called expansion microscopy, where they have this hydrogel material that can very uniformly expand. In principle, to like thousands of times the original volume.
So it's like physical magnification.
Physical magnification. And so Sam and I and other people who were kind of in and around the Boyden lab were very interested in using this physical magnification in combination with various other methods for neural connectomics. So we're mapping the wiring of the brain. So this is now not the activity, but it's the wiring. And part of the reason we were excited about that idea is that when you do this physical magnification, the brain tissue that's expanded in that way becomes optically transparent. So you can use an optical microscope, whereas the normal way of doing brain mapping involves an electron microscope in order to get the necessary spatial resolution to actually see the wires and the synapses between neurons. And this in turn requires very, very thin, like nanometer thin slicing, which makes it kind of hard to scale. The electrons are also kind of destructive of the tissue, makes it hard to interrogate the tissue multiple times and see what's happening with different molecules in it. So we were very excited about this optical approach, and Ed was very excited about this optical approach, and they've developed a whole series of papers of kind of breakthrough ability to do expansion microscopy of different kinds to do all sorts of bioimaging. But it also is becoming, I think, clear to both Sam and to myself and others involved in this, Andrew Payne, who ended up being one of the founders of this focused research organization called E11 Bio, that actually getting all of this stuff to work together to actually make connectomics a high throughput, scalable, repeatable method.
10-year process.
That's not like, that doesn't fit in like a postdoc project, right? That needs like something that looks like a deep tech startup level of like engineering team to build it. And yeah, there's a lot of, image analysis, automation, chemistry, sample processing, all these things. It's really like a cross-functional team that has to do this, and like ideally like a professionalized cross-functional team rather than like a individual kind of discovery-oriented, grad students and postdocs. And so that motivated Sam and I and others to sort of try to create non-academic centers where we could sort of spin off some of this work into a more industrialized mode of operation. And we failed at doing that in a few different contexts. But anyway, it was very much in our minds. And so Sam wrote this concept of focused research organization as I think it was the last chapter in his thesis. He was like, look, I've invented all these cool things. And by the way, kind of a postscript to my thesis, what we really need is like a focused research organization. And I had been working with him and other people to try to set up.
What year was this?
This was like 2019 or so. So I've been working with him and others to try to set up effectively what were focused research organizations.
The big validation for this, I don't know if you use this as an example, but like OpenAI, it's got to be, that's got to be the most validating thing, because basically since that thesis, now we have proof that you can produce this like economy changing, potentially.
Yeah, and isn't it funny that it's almost like they were able to make more progress than other AI companies by starting as a nonprofit?
Yeah.
It's kind of weird, and I think that we are starting to see other types of hints like that there's something about just the intentionality of going in with like, we're going to get these VC grade returns on this like 5-year time scale and make this very specific, adoptable product where we've really thought a lot about the business path for it, can actually kind of like suck some of the air out of the more fundamental technology development. And so, yeah, like early OpenAI, like maximize the engineering, maximize the presence of the talent there, build a platform where you could explore multiple different kinds of avenues of deep learning. And then ultimately, like Transformers, more like invented at Google, but they were able to kind of take off with that. So yeah, OpenAI is maybe not exactly early OpenAI, not exactly a focused research organization, but it's kind of like part of a sort of superset of the sort of nonprofit, engineering intensive, professionalized research labs.
Do you think the nonprofit aspect is critical, or is it the time horizon? And kind of like
I think there's a little bit of a fuzzy boundary, because even we talked about Neuralink, right? Yeah. I mean, that is kind of a for-profit company, but it's also not like narrowly channeling itself. Normally, when you do medical device, neuroscience, you're thinking about what's the exact disease indication. How do you make the device as simple and minimal as possible to get through the FDA? And Neuralink, of course, was a much more expansive kind of engineering, kind of build a fundamental platform technology, even if it takes a really long time, is really expensive, and you're kind of negative on your balance sheet for a really long time. So I think that there's you can have something that's sort of for-profit entity structure that looks a little bit like a FRO and what it does research-wise. You can have these things like OpenAI that were kind of in the early days, kind of clearly it's like very much a company now, but in the early days, I would call it like a professionalized, engineering heavy research lab or something like that. That's non-academic incentives. Anyway, so we ended up crystallizing, okay, well, if this is needed in connectomics, maybe this is also needed in lots of other fields. And for a variety of reasons, kind of in early 2020, I was stopping my previous job. It was the beginning of COVID. I was moving back to Massachusetts. I had been living in London for a couple of years, and I took on a fellowship with someone named Tom Khalil, who helped me get this fellowship. I had actually met him because he was one of the people involved in that Obama administration brain initiative that I mentioned. So with Tom then, and sort of building on the experience I had had with Sam and this crystallization of the focused research organization concept in Sam's thesis, we really tried to figure out like what that actually would be, what that could look like, and we went and interviewed lots and lots of scientists, not just in neuroscience, and said, okay, well, is there something you can't do in your academic lab? You kind of can't get the students and postdocs to do. It's not really the right incentive structure. It's too big in some way, engineering-wise. And also that you would really struggle to like do in your company or raise from VCs or so on. Are there any other things like that beyond connectomics? Mm-hmm. We had pretty strong hunch that there was, but that hunch has then been like very much confirmed, and so that is then the basis of Convergent Research as a way of spinning up those projects. We're sort of a builder and parent organization for those projects, and that has then made the FRO something that we're trying to put into practice. It's still early days of it, but we now have like 9 or 10 of them, depending on exactly how you count the stages of development, but of FROs that are up and running. Yeah.
Has anything you do actually been impacted by AI, like in a meaningful way?
I think that the connectomics field is being significantly impacted by AI.
Okay.
So what I was mentioning about E11 Bio and doing molecular barcoding and different labeling methods and stuff to basically reduce the amount of proofreading that's needed in neurons, that's going to be happening multiplicatively with major advances in AI, because there's actually an enormous amount of proofreading that you need to do a mouse brain or a human brain of all these microscopy
But what about
images.
just your personal work? Like, I feel like the promise of AI was we're going to be
writing internal company memos and stuff like that is definitely getting done. I'm also been trying to apply it to the E11 Bio. So there's another FRO that we haven't really talked about that has to do with mapping drug target interactions. I've been taking their data set that they've released online and trying to find drug repurposing opportunities in E11 Bio. So I'm sort of trying to do it.
But can you just tell ChatGPT and deep research, like, find me a FRO? Like, I want my next investment.
I think so far it needs a bit more prompting. It's also something that's happening hugely, is intersecting with what I was talking about with lean and math. But so far it hasn't been leading to a huge amount of like novel stuff that's like better than the ARPA-H program manager or whatever for scientific road mapping kind of things, but it is not so bad. So I actually did ask it like come up with a research program for like the molecular 3D printer or nanotechnology, and it's like it is better than like many people you would ask in like relevant contributing fields. Let's say like if you just ask a chemist who just is like amazing at organic chemistry, make a research program for molecular machines, they may not have been thinking about DNA origami, they may not have been thinking about these other things. And it actually, deep research actually came up with like okay stuff there. I'm optimistic that it will be possible for that to happen, and certainly that it will also take out lots of other kinds of work, you know, that's involved in the research process, and eventually close the loop with self-driving labs and so on. But it hasn't been the main driver.
But this is like a big
so
disappointment, I feel like, with AI, because it's hashed together this massive amount of knowledge, and yet it can't really come up with anything too insightful that like it's not insightful at a level that is superhuman or even as good as like you're the best expert in the field.
Yeah, and I've seen some discussion about this. Is like, is it sort of systematically worse than us at, kind of really like combining like latent reasoning? Like it has lots of knowledge, and can it like actually combine that from disparate domains to come up with something like really new? And asking the question of how good are we actually doing that, and what kinds of scaffoldings or circumstances are involved in us like just coming up with totally new insights. And there's the question about continual learning and data efficiency of learning. So on the pessimistic side, you could say, well, the brain has algorithms that are basically solving all of these things at the same time. The data efficiency problem, the continual learning problem, and the kind of like insightful like exploration or like new creative idea suggestion might all somehow be tied to some kind of like statistical algorithm that the brain uses that just isn't like backprop based, learning. And so that's sort of on the pessimistic side with LLMs, is like there's something fundamentally wrong. And on the optimistic side, like it takes a whole civilization actually to bootstrap itself to the point where you have creative stuff, and that's relying on focused exercises with particular texts and stuff that allow you to sort of notice those patterns. And kind of multi-agent systems is really how you actually get insights. I mean, I guess there's examples of high school students coming up with totally new math and stuff like that, but even they're like going through a very focused program of study that's kind of very specific curriculum and narrowing down what their focus is. That isn't just like, hey, I woke up this morning and I suddenly realized that this thing I learned and this thing I learned actually creates a totally new theorem or something, right? So it's hard to say,
Do you have a favorite FRO or something which impressed you in some way? Or?
It's hard to say a favorite. I've mentioned sort of already now 2 neuroscience examples. And I do think neuroscience is going to continue to deliver. Like everything in neuroscience, basically, I think is still like very much limited by technology, and a lot of the technology development is still limited by having this type of like focused structure to develop the technology. So I mentioned ultrasound-on-a-chip in the human brain. That's called Forest Neurotech, and E11 Bio. 10 to the 11 is the number of neurons in a human brain. So it's called E11 Bio, on this optical connectomics technology that builds out of the frustrations that Sam and Andrew Panning and I
and that's expansion microscopy
based connectomics, optical connectomics. Yeah. So that's one of the first ones. But one of the things that's
what
been
is
surprising,
that physically? It's like you take a brain out of a rat, you slice it, and then you expand the slices. Is that what it is? Or?
Yeah. So you fix the brain. You with chemical
fix is like you essentially polymerize the brain.
Yeah, you basically it becomes a kind of
like an
a
epoxy
block where the proteins can't move around within it. So you sort of you fix it, and then you label the neurons, and a lot of what E11 has been doing is about what is the strategy for labeling the neurons.
How do you basically such that you can see connections, or
so they
how do you even do that?
So part of what they've been working on. It's not all of it, and there's also a lot else going on in the field, and how do you just label the raw morphology of like where the cell membrane is going? That's just the shape of the cell. How do you label that and image that with expansion? A lot of that's been happening, and E11 is doing a lot of work on that. But one of the major things E11 has been doing is trying to put more information into the tissue so that you can then read it out later and disambiguate the cells that are next to each other. So they've actually been using viruses that put in a combination of different antigen. So they basically that they can label a given neuron. We'll get a random combination of protein markers in it, and so it's much more than just a mixture of, let's say, red, green, and blue to give you a mixture of different colors. You might have, let's say, 30 different labels, and any given neuron might say have 5 out of the 30, but 30 choose 5 actually starts to be a large number, and 40 choose 10, and et cetera. That starts to be a very large number of distinguishable labels. If you can individually read out, are each of these 30 proteins present or absent on a given sort of pixel of the image, that will tell you which combination of viruses infected that neuron, and therefore, and those combinations are ultimately like extremely unique across neurons. So you're basically digitally barcoding the neurons with different combinations of proteins. And so this molecular barcoding technology is something that they are about to publish a preprint on. How that then interacts with ultra high spatial resolution with the expansion, and what's the different combinations of this barcoding method and the morphological labeling and the imaging. Can you get to a point where it's actually just very easy to disambiguate which pixel basically belongs to which neuron with an optical microscope? So they're doing all of that. So neuroscience is just, I won't say a specific FRO, but I will say that the applicability of FROs to neuroscience is something that's been, I think, what we expected, but also exciting. And
wasn't there a failed thing like a FRO in Europe where they were going to model the entire brain in silico, and it didn't work out?
There was something called the Human Brain Project, which I classify as a mega project. You could argue that the U.S. Brain Initiative was also a little bit like a mega project, and so I think of mega project as like billions of dollars, thousands of people, rather than,
it's unfocused research organization.
Yeah, right. So most of the time when you get to billions of dollars, thousands of people, it is pretty unfocused, and maybe that's actually good. That you don't want to just put all your eggs in one basket. But the Human Brain Project was originally actually trying to be pretty focused. I think it suffered from the fact that it did not have this connectomic mapping capacity, so it had to make a lot of assumptions in trying to make this focused effort to make a digital simulation of a brain. And I think they got to the point of making like a rat cortical column or something like this, but they had to make a lot of assumptions about the connectivity and circuitry in that. So I think there were some scientific issues that maybe it wasn't quite ready to actually make a full neural circuit map without making a lot of assumptions about the statistics of how connections are made, and they don't know what the learning algorithms are and all this other things you might want to know. But what ultimately happened was a kind of political backlash. It was so much resources around this focus project that the rest of the neuroscience community sort of rebelled and said, actually, this should become an unfocused research organization. And so part of the reason we scoped the FROs to be like these finite duration, extremely specific, tool system or data set. Don't oversell it. Like this data set or tool should be extremely catalytic, but it's not equivalent to like solving all of neuroscience. So you have these very specific technologies. Just as no one would ever say the Hubble Space Telescope solves astronomy, right? It's like this is a mirror that's this much bigger. That's going to be very helpful. But we're not claiming that all of astrophysics is now all part of the Hubble Space Telescope project. It's just a useful tool. And so we've been actually, partly also trying to scope these as significantly larger than a kind of typical NIH funding of R01 grants is maybe a grad student and a postdoc and a professor for a few years, much larger than the kind of individual investigator typical grant size, but also much smaller and much more focused, much more specific about the deliverables than mega projects. Yeah.
So how do you find the funding for FROs in general? Because one paradigm is like a for-profit organizations. There's like investors, VC funded.
Yeah. And something that's great about the VC model is it does allow you to have this phase where you get enough capital to like really make a technical leap, ideally, right? It's like, and many of those projects will fail. Some of them will be incredibly successful, 1000x returns or something like that. But that statistics basically works out that deep tech investors can spend, $20 million something developing a technology if it has a chance of becoming, the next Waymo or something, right? And so that's a great model, but it doesn't work as well for like public goods creation, right? Think of the Hubble Space Telescope, like the market is astronomers, and they're all very happy, but they didn't really pay a meaningful
Also
amount of money.
long-term returns.
And also long-term. And so I would love to find ways to have more returns-bearing financial structures for this, but so far we've raised philanthropic grants, gifts, donations from 30-something different philanthropic donors. We've also gotten a certain number of government grants. We have a partnership with the Advanced Research and Invention Agency in the UK, which is probably going to fund a FRO, maybe two smaller FROs in the UK. We are working on trying to encourage other government agencies to create a sort of a FRO-compatible funding mechanism.
What percent of philanthropy goes to science? It's got to be like almost very low. Right?
Actually,
I know there's the Chan Zuckerberg
that
initiative.
number. I mean, I think this science philanthropy is already, even without anything is already a very meaningful and significant chunk. It differs in different fields, but hundreds of millions of dollars a year going into just even certain diseases like Parkinson's.
Yeah, but I feel like some of those are not 100% science. It's already like in the application, closer to the applications. And also, if you look at, I don't know what the entire philanthropy market is or whatever philanthropy, the chunk, there's also probably a lot of, people that would like to donate, but just don't believe that there'll be any outcome. If I model this myself, like if I, had the money to make very large donations, I wouldn't know who to give it to. Like, I'm not sure that if you just show up to Harvard and tell people, here's some money, like make the science, I'm not sure if anything would change.
I think the system is set up in a way that is actually very tricky on both of those ways, because if you think about it, if you imagine you're a high net worth individual, right, a lot of this is going to read to you as an exploit, right? Harvard wants your money, but so does Stanford, right? And how do you actually decide between Harvard and Stanford other than some sort of emotional vibes? And at the university level, they're going to want to kind of spread that wealth relatively equally among large departments or centers or schools or something like that. So if the philanthropist really just wants the one thing that's going to be the best technology for neuroscience or something, maybe they can try to work with the development office or work with the president of the university to find what are the things that Stanford is doing that are particularly good in neuroscience or could be done. But you're often not saying this is the exact deliverable. You're funding 1/3 of the Hubble Space Telescope, and it will be done in 8 years, and it'll be available to the entire community, right? And so part of what we're trying to do with the FROs is to get very concrete about the deliverables, timelines, et cetera, which is inherently a little bit tricky with science. I mean, there is technical risk in these projects, and things pivot, and so how do you set the expectations correctly? But we are trying to focus it on, like, this is the concrete bottleneck in research that you solve, and this is the chunk of it that you solve with this amount of money, rather than like donate to science generally or to a school. Although it's obviously also really important that people do donate to schools and to kind of much more diffuse, less outwardly legible process. Because the risk of, on the other hand, of doing something like focused research organizations saying, if you put in this money, you'll get this telescope, right? Great, but a lot of science is not really outwardly legible and predictable in anything like that way. And so it is actually really important, I think, that universities have endowments and that people donate to entire fields and in a very broad way, or support people rather than projects. But our focus has been in, like, if you put in this much money, here's like the best shot we can take at like this telescope or this microscope or this.
And is that
tool,
typically also, you mentioned people, I feel like in VC funding, people are really critical. So it's often, especially early, people are looking at the team.
Yeah.
Is that something that, like, when you look at these projects, like, to what degree are you thinking, like, what's feasible versus where are the people available to actually
It's a huge factor. It's a huge factor.
Happen?
And I have been positively surprised. One of the major critiques that we faced, let's say 2020, going in around talking about this was, okay, but the top scientists aren't going to want to like join a FRO because it doesn't offer tenure, it doesn't offer the chance at being extremely wealthy over that relatively short period. You can potentially spin out companies from FROs and so on, but basically depending on whether that's the right thing to do. But it's not like a VC-backed company where, like, if it's successful, you're like Mark Zuckerberg or something, right? And it's also not the stability of tenure or the stability of a big company. And so people were saying, okay, well, great, people join this. I think we have been very pleasantly, I guess, whether surprised or confirmed, depending on sort of how we thought about it at the beginning, but fantastic scientists, leaders are joining these things. That being said, it's a lot to ask on a project founder, just as it is with a VC-backed company, because there's a management component, and there's a salesmanship component, and there's a figuring out what happens after the FRO, and there's all the technical work that has to get done. And scientists are not necessarily trained very much to actually build organizations. So both of you are seeing in sort of deep tech companies that a lot of the issues are like people issues. And so there's a lot. So we have the founders get executive coaches. There's an element of selection of some people just really want to kind of be doing research and don't want to be managing a team. Do you want to do that? Are you trainable to do it? Do you have experience doing it? So the founders are a significant component, and of course the scientists that join are a significant component. But I would still say that on net we're still funding limited, rather than we have more ideas that come along with good founders than we have funding. But eventually we would come to a point where we have lots of ideas, and then we become founder limited. And I think it's a while before we're completely out of ideas. There's just not things that fit the model of well-defined tool systems, data sets, catalytic techniques.
Yeah, I guess it's kind of like, you had all these discoveries that were kind of like new physics almost. I mean, for example, X-rays, like one guy discovers X-rays in his basement with basically a light bulb. And then 2 months later, people are X-raying, like doctors are X-raying people.
Yes.
And Neuralink is the exact opposite of that. It's like, okay, we're hiring 100 people.
Right.
And we're going to hammer this. Like basically if somebody in 1900 said, or I forget when it was invented, around 1900, like before X-rays were found, said, we're going to find a way to image bones.
Right.
Yeah. We're going to get together 500 people, billionaire backing or whatever. So, I mean, maybe things like that used to happen, but it feels maybe this is like a symptom of a lot of the low-hanging fruit being gone, that we need to get together these big groups of people to make progress.
Yeah, it's a little hard to say. And I think in some of these things that happen over decades, I mean, eventually MRI, nuclear magnetic resonance became MRI, became functional MRI. Actually, I think some of the early fMRI stuff was partly as a result of stuff happening at Bell Labs and things like that. So there's some question about organizational structures even there. But that also took decades, right? And if you want to be like, okay, right now I want to get on the path, the optimal brain mapping project, it just may not look like what the individual distributed self-organizing field of neuroscience has.
I mean, I think in science, we're still trying to, everyone is chasing that high.
Yeah.
Everybody wants to discover the X-ray in a light bulb.
Yes.
For sure, everyone's operating on that. I mean, I think maybe at least when they start out, right?
Physicists definitely are.
When you get into
right?
science, people are thinking that way early on.
It's like an old pattern, or the old recipe instead of trying to come up with.
Well, it's just when you read books about science. I mean, if you're growing up reading about science, you're not reading about project management, right? You're reading about.
Oh, sure, yeah.
You
want this thing that easily affects all the other areas.
Yeah, or just it's more about insight and kind of really amazing experimental design and things like that. Yeah. You don't really. I mean, maybe the Manhattan Project is one exception where you read about management being part of the the magic, right? And kind of organizing humans versus a single person turning the problem around in a way that's just incredible. I remember the way they found what alpha particles were is they basically just put one glass bulb into another and put the radioactive substance into one, and then it made a vacuum outside, and so the alpha particles eventually collected between the two.
Yeah.
And this was Rutherford did this with a grad student,
so
on
cool,
a weekend or whatever, right?
Sorry, no more problems of that scale.
Yeah, it's hard to say. I mean, I think that I would hope to think that there are still there's a huge amount of role for the individual creative, and also for just exploring these fertile fields, and then you have these transformational discoveries. It's not clear to me whether it's across all of science, but it's definitely clear that there are some problems that are really limited by the social organization. Can we get together the team to build the tool that someone else uses? And that building the tool may not be the glamorous part of the discovery, but it is. I mean, I think, again, the Hubble Space Telescope, I think, is a great example of this, and maybe you don't actually read that much about those things in the history of science. That I certainly, when I was a kid growing up, I was interested in the big technology platforms in engineering, but I assumed that like the government sort of just like had all that organizational stuff like figured out. Like there was some sort of federation of people who would make sure that like the next Hubble Space Telescope definitely will get built. The resources will be there. The management will be there. The interesting thing is to just get really smart and like understand the ideas and be able to come up with better ideas, better experiments. And that's what I assumed. And I think that was a really great assumption to make because it caused me to like go and read a lot of books and learn a lot of object level stuff. But at this stage of my career, I'm like much more like feeling that things are limited by human dynamics, organization, funding, resource allocation, project prioritization, those types of things that are kind of boring, but yeah.
So I guess when you were working on chip fabrication with DNA, back then there was a lot of kind of like runway for normal lithography. But although everyone's always predicting that it'll end, but I mean, we're getting there, I think, with EUV and, it's traditional lithography is kind of hitting roughly its limitations, in terms of like order of magnitude. What do you think? Is there like, first of all, how was that supposed to work? Like, what was the vision there, or like, what's the vision in that field? And what do you think? Like, is there a possibility that it really does become important in the next? Like,
I think there are
20 years
things, and there may even be that there could be sort of FRO things that a role for FROs in this on some things that would take you sort of meaningfully beyond silicon. And I'm not an expert on this, but people talk about 2D materials. Graphene being a classic example, but there are many others of sort of 2D materials that have kind of fundamentally different like transport properties. Can you make things with discrete components like carbon nanotubes? So that's one category of thing. There's and kind of how do you do sort of 3D integration? There was a great group of people, mostly out of Stanford, that talked a few years ago about sort of 3D integration with carbon nanotubes, graphene nanoribbons, and sort of 3D stacks of those and memory elements included in it. That's kind of really beyond silicon. And there's like one foundry called Skywater, I think it's called, that has been able to have some ability to do that. There's actually a group at MIT has worked on some of that stuff. So I think that these like discrete components in 2D materials is pretty interesting. I think that superconductors outside of quantum is pretty interesting. So we've talked to this guy Jeff Shainleen, who has a company now called Great Sky, but they're using Josephson junctions and actually optical linkages between them, not for quantum computing, but for kind of classical sort of neuromorphic style computing. So there's definitely stuff in like the beyond silicon world that I think might need this type of like focused engineering research push. But some of it is also, are the foundries compatible with this, and manufacturing and getting big companies to invest.
Has there been like a biologically produced transistor or something like that? Like where you...
There's been some stuff like that. You can even do some stuff just with DNA. There's
DNA transistors?
Molecular
or just like enzymes, etching.
There's also an intersection with biosensing, where sort of you have molecular electronics on a chip and a transistor that's sort of gated by something that's binding to a molecule or stuff like that. So there you may know more about this, a lot more about this than I do, but there is stuff there. I'm not sure that, this is why I would describe it more as a research field, because I do think there's a big gap between any kind of like polymer-based protein or other system and like just all the material properties that we have with silicon and the way to just fabricate it in this very bulk scale. So I think of the nanotechnology kind of artificial ribosomes and stuff as kind of like, it's very exploratory research field. And in my ideal world of science, we would have like these DARPA level investments, even in things that are extremely exploratory. And I'm not exactly sure what device I'm going to make with that thing. In the real world, these things are pretty expensive, and so some of the more practical questions are like, can some of these big companies, Applied Materials or stuff like that, start making pushes in things like 2D semiconductors and sort of making almost like the supply chain for things that aren't just silicon, basically. But
I mean, if you pull apart like a insect or something, right? All the functions that the insect is doing across many different orthogonal axes are better than anything we have.
Right.
Yeah. So it's like energy density and storage, just the ability to self-heal. Like you just start listing all these things, right? And it feels like, are any of those functions, are those potential FROs in and of themselves? Like basically saying, all right, here are all the functions of life, right? That we've seen that are, I don't know, 100 times better than anything we have.
Yeah.
How do we generate this?
Can we make a micro robot?
Or even just one component of it, like can we get like basically something that's powered by that has the battery life of an ant, right? Like I mean, it's a funny thing to say, but I mean, if you look at the battery life on insects, it's
so efficient,
crazy.
But the same about the brain, right? Look at how much power our brain absorbs.
I mean, you can these examples are like they're littered, right? Like you can just pick them up with a shovel, like from any
and something
system.
I haven't totally understood, honestly, is how much. So the neurons, they switch very slowly, and I think there's some quadratic dependencies in there if you're allowing yourself to slow down. So if you're in terms of the algorithm, it allows you to like do slower processing and rely more on memory rather than more on compute. Like so partly is like architectural, is like it's kind of relying on accessing memories more than it's like doing a lot of like cyclical real-time computing. Part of it is the substrate, right? Is it's just like it's charging up voltages across the cell membrane, but they may be operating in a noisier regime where it's able to be a little bit more stochastic and not burning all the other kind of leakages and operating at smaller voltages than transistors are. So across the membrane, it's kind of like tens of millivolts range versus kind of volt scale. So there's some aspects about the voltages you're using, and there's some aspects about using chemistry, and there's some aspects about being stochastic, and some aspects about the architecture. I would like to really understand what is it exactly, why is it that the brain is so efficient? It's like insanely efficient, right? People are talking about how to power data centers with nuclear power plants for things, GPT, 7 or whatever. And
it's like, maybe humans are only capable to do energy non-efficient technologies like Bitcoin, GPT, electronics.
Yeah, and so how do you this kind of thermodynamic computing, or can you use stochastic algorithms and build the stochasticity into the hardware? Is that then more compatible with switches that operate at lower voltages and stuff like that? So there's all these things. I don't have a full analysis of it, but I think that there's multiple things coming together. So there might be multiple FROs. I want to get the connectome to understand the architecture, and I want to maybe have low voltage transistors, and so.
How do you, I mean, this is more of a meta question, but like obviously in this role you've basically created for yourself, right? You kind of, at least nominally, should know a lot about a lot of different areas of science, right? And like kind of figure out where the edge is in a lot of different areas. What's your strategy?
At
like
kind
how
of
do you
a crude
actually
level? I'm just reading all the time, talking to people. I think we get a lot of value out of other people who also kind of have this like programmatic perspectives on research. So former DARPA or ARPA-E, program managers tend to have this like systems engineering kind of view of things. Sometimes people from industry actually have kind of more of like a cross-disciplinary view. Sometimes you're talking to academics who have very specific knowledge in some area, or come from more of like a physics background or some unusual background that they're bringing to some field. So we try to talk to a lot of weird people, and then ideally Convergent's mental map is kind of like weird aggregate of lots of other weird mental maps. It's not just like going directly from like this grad student making this discovery all the way up to like what projects do we do. It's like this grad student making this discovery makes it to what the ARPA-E program manager's thesis was, which makes it up to how we're thinking about that in comparison to what this other thing is, which then filters up to us. So we're kind of like a mixture of experts model and rely a lot on networks also. But also over time, as you just hear these conversations, if you just ask different questions, right, sometimes it's pretty easy. It's just like, tell me what would be your equivalent of the Hubble Space Telescope in your field. And people would be, I haven't really thought about that before, or I haven't written a grant on that. I've thought about it, but I haven't spent time. Okay, well, I'll spend like a day thinking about that and I come back with a 3-page document about what the Hubble Space Telescope is in their field. I didn't need to do anything. I just need to ask a question that sets them off on a kind of ambitious direction, and they come back with something great. So
you're kind of at the interface, maybe this like gray area between VC funding and traditional science funding. If you compare the Boston community, which is more traditional science and pharma, versus SF, which is really has its roots in like go-go software type, and before that chips type funding. What do you think of these communities? And kind of, is there something that Boston can learn from SF and vice versa?
That's more how I would think about it, is sort of on average things to learn from each other. So, I mean, if you think about stuff that Boston has produced, like Moderna, wow. Or even if you say, well, I mean, I think traditionally in like the bio space, Boston maybe has been more like heavily invested in certain forms of risk reduction that I think they can take, that culture can take to an overkill level, right? So if you're running a therapeutics company, you can only have as the CEO of that company someone who has already, run 5 therapeutics companies, right? Raised over this much money, et cetera. I think that there's kind of an inside baseball aspect to that, which may sort of select for navigating the pharma industry or something like that as it exists. And like narrowly steering towards certain kinds of exits, but not like revolutionizing technology. So like on the one hand, like that conservatism of like Boston biomedical culture, I think has some problems, in that the Silicon Valley culture of like, yeah, what if we just made this like crazy new chip thing that would let us develop, thousands of new drug targets or something like that, build platforms. That's kind of closer to how we think with the FROs, with platform technologies. But if you actually look at some of what's actually happening, first of all, I live in Boston, have lived in Boston for a long time, very much come out of the academic culture in Boston. I sometimes think the academic culture is more risk-taking on average than what you see in Silicon Valley startups, if you look at kind of the depths of MIT.
Did you consider a move to the West Coast?
A little bit, yeah. I'm mostly here for kind of family stuff, but it also kind of works well. We have two of our FROs are here, 2 of our FROs are on the West Coast, and we have some that are kind of more virtual remote. We have one that's in Raleigh, North Carolina, one that's in New York City. So some that are emerging in the UK and London. I actually really like London. I'm bullish on London for a variety of reasons, heavily to do with combination of ARIA over there and DeepMind and a few other things going on. But on average, like I think taking risks on true platform technologies and not over-defining, taking risks on crazy or less experienced founders, younger, more close to the science as opposed to close to the business of running a sort of biopharmaceutical enterprise. That's all good stuff. But also, if you look at what's coming out of Boston, I mean, think about something like Moderna, right? Think about even the fact that like Ginkgo is kind of a complicated example, but like is in Boston, although they went through Y Combinator to kind of have their inflection of like investor interest. Lila Sciences, another flagship company, is doing way more on self-driving labs, as far as I can tell, than like Silicon Valley is doing so far. There's a lot of talk about AI for science, but Lila Sciences like hired all the self-driving lab people in Boston. So I think it's complicated, but we're sort of this orthogonal category. I think you could say that we're part of the founder-led movement or something like that. The Pillar and Petrie and some of these other VC firms have Y Combinator sort of one of the sort of scientific founders. I think we're kind of in the scientific founder-driven category rather than like established business person category. But gosh, you look at the actual track record. I mean,
but biotech isn't like a multi-year downturn.
Yeah,
and like I guess it wouldn't be a bad thing for you if that could be attributed to a lack of ambitious projects driven by small teams with young people. Like, I do wonder if like they had been doing FRO type work for decades leading up to this, would we be in the same situation?
I think there's, it's hard to say. I mean, there's a lot of really good work right now that's struggling and not getting funded because of this like biotech downturn. I don't entirely understand the causes of it. You look at public market, biotech is not doing super great, and so you can say, does that filter back into how many IPOs and stuff there will be? Yeah. And but then we're coming out of a period where we have these totally revolutionary, mRNA, CRISPR, cell therapies, perturbational screening and target finding, multiplex with single cell stuff. So it's like technologically, like even without any FROs, the basis set technologically for doing incredible stuff in biotech seems to be there. And it's like, I'm frustrated that the markets are not repaying people essentially for maybe investments they made into, maybe 2021 was over exuberant on certain things as to whether it fits the VC model, but technologically I think there's a lot of good stuff there. So maybe there's some 2021 stuff that in 2025 would have been more FRO-like, but yeah.
What do you think of Hair DAO? I bought some coins in this thing called Hair DAO, which is like a coin thing where they're rubbing different drugs on their hair to try to like regrow hair. Have you heard of this?
I haven't seen this much in detail. The one that I followed, I mean, I've been interested in, I mean, I read the Bitcoin and Ethereum white papers back in the day and stuff like that. I've been kind of interested in this stuff. We haven't really done any decentralized science kind of stuff directly in Convergent. We've just been getting like traditional grants, just write us a check with US dollars, you know. But it was pretty cool seeing this quantum biology DAO actually managed to raise enough money off of a DAO to start like a meaningful kind of small to medium sized quantum biology FRO, where they're trying to make microscopes to look at spin states in biological systems. And so I'm optimistic the decentralized science stuff can kind of do something. Obviously, it tends to a little bit exuberance on like 10 dimensions simultaneously, where the hair growth is going to work and we're going to fund it with crypto and all this stuff. We're going to make lots of money. And like,
the thing that attracted me to that one, and I'm like a super skeptic actually of DAOs. I don't even understand. I honestly like, you know, and I'm like,
it's a good market, right?
Been interested in Bitcoin,
aging,
you know, very long
yeah.
time, and I somehow don't understand DAOs. But basically, what I liked about them is that they had just like a Discord with people rubbing drugs on their head.
That's cool. Yeah.
And I was like, that's new. You know, that's interesting. They've got all these people rubbing these drugs on their head, and they have like a camera system that they give you to look at the number of hairs that have like per square inch. So
that's
they're doing like
cool.
a quantitative.
Yeah.
It's pretty interesting. I mean, to make a study like that in traditional pharma would be, you know, you'd have to get the guy who's had 7 exits in pharma, and he would hire 7 VPs. Yeah. And those guys would hire a CRO that would recruit patients. You know, I mean, by the time you have someone rubbing drugs on their head, it's like $100 million enterprise, right?
I do think that's pretty interesting. Yeah. And how can we get more data in a more distributed way? I mean, Patrick Carlson actually just had a fun tweet about Reddit, and basically Reddit forums are essentially like the best information there is on like how patients respond to different chronic disease interventions, right? You have some sort of chronic gastrointestinal or other type of problem, and you try different things, and it's really Reddit where you have like this fine-grained information about what people tried, what worked, what didn't, and that is way different than patient recruitment in clinical trials. There are things you can fine-grained level of information that you probably get access to, and that so of course a lot of it is like correlational or biased.
Yeah, it's like anecdotal.
Like how do you actually do that? So we're actually going to add that Twitter, a bunch of things linked in that Twitter thread to GapMap.
Okay.
Sort of patient-reported trials or something like that, or anyway. Yeah.
Related to these DAOs is like, I think crypto somehow, unclear how, but really relates to longevity somehow, at least in the people that are interested in it. What's your take on whether there are interesting problems in longevity that are labile to like a research organization? And specifically, I think you mentioned cryonics as like one that you're excited about.
I definitely think that there is throwable and other major low-hanging research project fruit in longevity, especially if you're willing to operate at that research platform, research enablement level versus making a bet on a therapeutic, right? I'm on the board of Norn Group, which is a longevity-focused nonprofit that sort of does similar type of kind of scientific road mapping, if you will, gap mapping, et cetera, in longevity. And they recently came up with something called the Clock Initiative or the Clock Project, which is basically about validating different types of aging clocks, right? There's lots of aging clocks that say this correlates with your biological age, and then separately there's people that do interventional studies and say, if I give you this much rapamycin in male mice versus female mice, et cetera, you know, what's the effect on lifespan? And then there's sort of separately things that do kind of small studies. So the NIH interventions testing program sort of does this systematic testing about interventions and sort of statistically valid, reproducible effects on lifespan. Then there's these people making clocks, and then there's sort of separately people doing kind of like really deep omics. Like CZI funded this thing called the tabula muris CNIS, which was like single cell sequencing and sort of cell states as they evolve over aging, cell type populations in different tissues as it evolves over aging. I don't think those should all be separate. So this clock validation project is like, okay, can I combine the epigenetic clocks and see if they actually predict causally, right? I do an intervention, my clock changes. Does that then affect my ultimate measurement of lifespan, and do that in like a really rigorous way with like the right genetic background of mice, where you're not just overfitting to a certain genetic background? And then I would like to combine that with lots of omics to actually see what's the physiology that's changing. So I basically think there's lots of like aging sort of perturbation plus clock plus lifespan plus omics. That is like, it's a big project to do that. And it can seem like kind of a fishing expedition, but I actually think we don't understand like the multi-system kind of physiology. If I do, is one aging drug operating by amplifying the same molecular pathways as another aging drug, or is it actually operating in two totally different tissues, which then synergize at a physiology level? So I think that there actually should be like significant amount of like causal aging omics and clock making that's like just more grounded, and probably requires these like large projects. That's just one of many things probably in aging that one could do. And then I'm actually really excited about vitrifying, forming vitreous ice, freezing without ice crystals of large organs, or maybe eventually entire bodies. There's a company, Laura Deming's company, Until, which is working on this. And also there's actually an NSF Engineering Research Center at the University of Minnesota, I believe, about sort of stopping biological time, where basically this is a problem of how do you very rapidly heat and very rapidly cool a three-dimensional volume of tissue, where things that you have access to, and you want to do that without forming ice crystals. So if you can heat it fast enough and you can cool it fast enough and then heat it fast enough, then you can do that without the ice ever having time to form large crystals.
Is there a physics limitation to that? I feel like, how do you overcome just the physics of heat transfer?
Yeah, well, I think something you have access to, then the question is, how do you utilize this? Is you have access to the fact that the bloodstream basically goes close to every cell. So can you use the bloodstream to load cryoprotectants into the tissue? Can you have cryoprotectants that stay in the bloodstream? Can you have, what the group at Minnesota does is has nanoparticles that allow the warming to be faster, where you have sort of like microwaves that can penetrate well, but then they locally heat these nanoparticles, which then heat the tissue. So you don't necessarily have to doing all the cooling just from the outside of this brick into the middle, which, yes, that takes a lot of time to propagate, but you can use the vasculature, you can use nanoparticles, you can use cryoprotectants. And so it's not obvious to me that has a limit, that it just can't go beyond, the certain size, right? Because we do have radiation that penetrates, and the vasculature does penetrate.
Can you cool something just to the point of almost freezing and then drop it in liquid nitrogen or something like basically so that there's like a wave of freezing that propagates very quickly?
Yeah,
I don't know if you can survive.
Yeah, there's a question of whether when you
survive?
do that, are you then forming the ice crystals, or are you not forming the ice crystals? But I think that vitrification of large volumes is not crazy, and of course we know that vitrification of like a few cells
works
if you do IVF or something like that, or you can do that for like small embryos or stuff like that. So
what's the next level up? So if you have embryo here, what's the next smallest thing?
Yeah, I mean, I think that there's some. I'm not sure that C. elegans is like necessarily the exact right thing.
I just mean in the body. Like, is it like a hair follicle, or like what's the next?
In terms of useful things,
Yeah.
I'm not actually sure exactly what is the next like useful size organ that people want to preserve. People really want to preserve things that get transplanted, hearts and kidneys and stuff like that. Yeah.
So looking back, like, who would you change anything in your life? I don't know, particularly, would you like go to grad school? Pursue a PhD, or there's like other paths you would go?
I think I probably would still go to grad school. Actually, for all this emphasis on new, team-based research structures and FROs and large projects and stuff, I think that there is something kind of irreplaceable about an environment that provides for like deep mentoring and thought transfer. Like the ideal PhD, right? You're basically mind cloning with variation, an advisor who's like incentivized to like help you develop like a new pattern of thought that like they might be one of only 5 people in the world that can like have that pattern of thought, or only one person in the world. So, and in a weird way, like the church lab, although it's this more like distributed process of hundreds of people versus like hours and hours at the whiteboard with George, I feel like I incepted like some different culture or like way of seeing the world. So I'm actually like very pro getting PhDs, and also very pro people just like mucking around in the lab and like trying stuff. We see a lot of people who are like really young students and they're like, I want to get involved in meta science and I want to get involved in creating FROs or companies, and that's really new exciting thing. But it's kind of like, yeah, that undergrad like kind of needs to like go and like learn physics before they're going to be able to like, even if they don't contribute to the Hubble Space Telescope, they might be the astronomer that uses the Hubble Space Telescope, and they might go in some other field. So so I'm actually pretty pro PhDs, with all the problems that the PIs are facing about how they get grant funding in the current system and all that stuff to maintain.
How would you, if you were getting into science today, if you're 20, how would you strategize? Like, where did which field? Like, how do you get to the edge of a field? Like, which field actually is kind of like gonna have some movement versus others? And
it's kind of silly, but I think the physics major was still great. It's a really great combination of formal and mathematical and abstract with kind of concrete, and how does it actually ground out in the physical world. So I've talked to Ed Boyden a lot about this too, but sort of training that combines some amount of like theoretical work and some amount of more like empirical work. Like sometimes you see pure mathematicians who come into the bio lab and they're just like, they don't understand the kind of logic of like how discoveries are made in the bio lab. And sometimes you see people who are biologists who sort of don't have this like abstract framework of like understanding physics or chemistry or computer science. And so if you have some training in the more empirical field that's like fundamentally just like about throughput of experiments and quality of experiments, and you have some amount of, and like a lot of like weird hands-on illegible stuff, tacit knowledge, and you have some training in like really analytical fields, math, physics, computer science, I think that can be really powerful as a combination for getting started in like our current world still. So I probably would still be pro like doing the physics major, and then maybe taking some biolab classes or stuff like that, doing some research.
Just one last question before we wrap up here. So you must, the same way that like physicists will get a lot of like emails with great ideas about like theories of everything, you must get a lot of emails with great ideas for FROs, right? So what makes a good, like if somebody's thinking about writing you an email for, I have a great idea for a FRO, what is a good idea for a FRO versus a naive idea for a FRO?
I think we want you to understand the context in the field of what is sort of what is going to happen in the field as a result of lots of great work, but with the existing structures, versus what counterfactually actually really needs the FRO. So we get some of these emails that are like, I have a project. It's just my project. It should be a FRO because I can't get funding for it. That's not really right. We get some that are things that seem like they're kind of already happening. We get some that seem like they're just like not very important if you solve them. But if you're working on something like really important, the area is really important. The knowledge you would get out is really important. And we want to show that you can sort of work on carving out the FRO-shaped problem. Here's what the academic labs are going to do if the FRO is actually produced. Here's the work that actually needs to live in the FRO. Would this be better as a consortium? And we can help people to some extent with thinking through this and talking through this. But like, we get a lot of things that seem more like initiatives or high-level goals or something being broadly important, or kind of boil the ocean, versus like the FRO will make this very specific tool system or data set. This is why I couldn't do that, even if I had a well-funded consortium of research groups in the existing institutions. Because a lot of things can be done in the existing institutions, right? So
it's in the style of like, we want to take 100,000 rats and robotically slice all of them, and then this data set will enable.
And it's like, well, tell me, are you sure you couldn't do the 100,000 robotic slicing machines in the academic lab? It depends, right? If it's a computer simulation, you could maybe just do that in the academic lab. If it's C. elegans, maybe you could do that in the academic lab. If it's really mice, and you really need the connectome of every one, now we have something that's like impossible, or we can't do it yet. Okay, what's the right shape and size where you actually do need that 20 or 30 person cross-functional team? Why do you need that team as opposed to another team? So getting the FRO shape right is part of it, and part of it is, are we targeting these like sufficiently fundamental gaps? We also see a lot of things that maybe look like they're ultimately aiming to make a commercial product or service, and so really the question is how to like do this as a startup versus just assuming that this is a FRO. So we also don't want things that are just like bad startups. We want things that are bad startups because they create public goods that are highly catalytic to science but not investable, not bad startups because they're just like startups but on the lower end of startups, right? So, yeah.
Is there where can people find you? Are you hiring for people to help you do what you're doing? Is there anything else people should know?
Convergentresearch.org. We have open positions. If you go to the Get Involved page, both within the FROs and on our core team of various kinds. And as this grows, we're going to need people to like help mentor and kind of sit on boards of the FROs and kind of be like entrepreneurial mentors for the FROs, and kind of the way like the Activate Fellowship or something like that might have mentors. All sorts of operational support that we provide to the FROs on our team as well. And when we're lucky, when we have enough funding, or when we really go after an area, we are looking for people to have want to spend time doing the scientific road mapping thing, go and figure out what are the FROs in some new area, what are the really deepen that gap map and opportunity setting, kind of like a slightly related skill set to what an ARPA agency program officer might be looking for. We're not always looking for that much of that because we tend to be a little bit more like funding and execution limited than idea limited. But in certain areas, at certain times, we try to roadmap a field, and so that's where more like scientists might get involved in the Convergent core team. But then there's lots of, you could either propose a FRO or you could join an existing FRO where there's like an incredible amount of cool science going on. So, yeah.
Awesome.
Fantastic.
Thanks a lot.
Yeah. Thanks for doing this.
Thank you guys. That was really fun. I really appreciate the breadth of questions and getting into some actual science.